live, and before Dr. Tamara's remarks, I do want to introduce her. She's joining us all the way from Europe. She has a degree in chemistry and PhD in biotechnology on developmental genetics from the University of Ghent, which is in Belgium. She's the co-founder of Orzyzon Genomics back in 2000 and was the acting chief scientific officer until the beginning of this year. She has been a member of Oryzon's board of directors since its foundation, and it's currently its first vice chair. She also presides the company's scientific advisory board since November 2020, and in addition, she's the managing director of Mendelian Life Sciences, a member of the scientific review board of the Alzheimer's Drug Discovery Foundation since 2016. She's also collaborated with Lakeisha in his mentoring program for new entrepreneurs since September 1st, 2015. As well, she has led countless scientific programs in Oryzon and the developmental of their LSD1 inhibitors from target validation drug and early stage drug discovery all the way to clinical translation. So, welcome.
Well, thank you, Sam. I'm very happy to be here. This is my second try. Yesterday went all wrong, and I hope that this time you will be able to enjoy the presentation, and I will answer your questions afterwards.
Good morning, everybody. Thank you for this kind introduction, and thanks to the organizers of this meeting to invite Oryzon to present. I will be talking about the pioneering of personalized medicine in epigenetics, which is a strategy which we apply both to oncology and to CNS disease. Orzyzon Genomics is a public company. This is our legal notice. We like to see ourselves as an epigenetic champion determined to bring new therapies to patients. Our epigenetics expertise is applied to oncology and CNS indications. We have 2 molecules with positive clinical data in human programs in phase 2. We have a leading and growing epigenetic platform with an expanding pipeline. We are one of the most liquid biotech stocks in Europe. We have cash and cash equivalents of around $50 million by the end of 2020, and we have cash runway till 2023. We have multiple shots on goal to address significant unmet medical needs. First one is yadadamstat, an LSD1 inhibitor envisaged for oncology, which induces differentiation and targets cancer stemness. The second is vafidemstat, a second LSD1 inhibitor envisaged for treatment of CNS disease and COVID-19, which has prosynaptic and anti-neuroinflammatory characteristics. And we have a growing pipeline. We have an HDAC6 inhibitor and other epigenetic targets. The HDAC6 inhibitor is ready for nomination, and additional leads against a variety of new targets are under development. But let's go back to yadadamstat, our phase 2 clinical stage asset in oncology. Yadadamstat is the most potent LSD1 inhibitor being tested in clinical trials. LSD1 is involved in different cancers and in cancer stemness. High levels of LSD1 often correlate with more aggressive forms of cancer and or bad prognosis. Yadadamstat is a highly potent and selective orally bioavailable LSD1 inhibitor. It's first in class, best in class. The profile of the molecule has been published in Cancer Cell, and everybody can go and compare the characteristics of our compound with other LSD1 inhibitors in the clinic. So yadadamstat proved safe, well tolerated in phase 1 and various phase 2 studies. Around 100 subjects have been dosed, and it produced strong differentiation and showed preliminary anti-leukemic activity in a first-in-man phase 1b trial in relapsed and refractory AML. So how does yadadamstat work? Yadadamstat binds to the FAT cofactor of LSD1, and this inhibits the enzyme's enzymatic activity completely and irreversibly. In addition, The occupation of the active site by yadadamstat inhibits the recruitment of LSD1 by a number of transcription factors which have been shown to be key in AML or in small cell lung cancer. Yadadamstat was the first selective LSD1 inhibitor to enter clinical trials. We performed the first in-human phase 1 study of yadadamstat in relapsed or refractory acute myeloid leukemia. 41 patients were recruited, 27 of which were included in the dose escalation and 14 in an extension cohort. The compound showed good safety profile and good pharmacology, and importantly, we were finding strong evidence both at the molecular level and at the morphological level of differentiation and the preliminary anti-leukemic activity. The results of this study have been published in the Journal of Clinical Oncology. I think it is generally accepted that it would be difficult to beat AML with a single agent. So we designed a next trial, ALICE, a phase 2 trial in AML with an LSD1 inhibitor in combination with azacitidine in the elderly or unfit. This was a multi-center, single-arm, open-label study, up to 36 patients to be enrolled. Primary endpoint was safety and tolerability, and secondary endpoints response, time to response, duration of response, and overall survival. The study is still ongoing, so the accrual status as per December 31st were that 21 patients were enrolled, of which 20 were enrolled as per protocol and 40 evaluable. So what we can already see in ALICE is that we are getting high ORR rates, rapid onset of action, and clinically meaningful durability. So 85.7% of ORR in 34 days, times to response, 58% of CR or CRI rate, and 86% of which show a durable response. Also, in those patients with more than 120 days on treatment, 40% were becoming transfusion independent. So this is very encouraging. The combo appeared safe and well tolerated. From 41 serious adverse events, only 2 have been considered to be probably related to yadadamstat, 1 intracranial hemorrhage and 1 differentiation syndrome. No QTc prolongation, no neuronal, hepatic, renal, or any other organ toxicity has been observed. The next indication of interest for treatment with yadadamstat is small cell lung cancer, a very aggressive disease which represents about 10 to 15% of all lung cancers, and in which LSD1 is a well-characterized target because NSA1, a transcription factor which is key in small cell lung cancer, is recruiting LSD1 to repress gene expression. So we previously showed that yadadamstat produces complete and durable tumor regression in different chemo-resistant SCLC PDX models. Yadadamstat was also efficacious in combo with platinum etoposide and other agents as ECIs. And we can say we identified and patented biomarkers that differentiate SCLC tumors by sensitivity to LSD1 inhibition. 2 studies were done with yadadamstat in small cell lung cancer, a phase 1 study in which the compound was used as a monotreatment, and a phase 2A study called clepsydra, in which the compound was administered in combination with platinum and etoposide. So this study was an open-label, multi-center phase 2A study. Biomarkers selected sensitive platinum 2L-ED SCLC patients, and YADA was used in combination with 4 to 6 cycles of carboplatin etoposide. And after the chemo, YADA could be continued to be administered alone. 14 patients were enrolled, and the study is finalized. So This combination was more tricky. The platinum etoposide in combo with yadadamstat showed hematotoxicity, while the continuation of treatment of yada alone was safe and well tolerated, so the toxicity disappeared when treated with yada alone. Despite this suboptimal dosing, efficacy signals were encouraging, with about 40% ORs and a mean DOR of 4.5 months. There was a 60% clinical benefit rate, and with full partial responses and 2 long-term stable diseases. One patient had 50 cycles in monotherapy after the combo treatment, with a total tumor size reduction of 90% and a relative tumor size reduction during yadadamstat monotherapy of 53%. So this treatment was definitely helping the patient. Paclitaxel is our phase 2 compound for the treatment of CNS disease. Paclitaxel is a small molecule LSD1 inhibitor optimized for CNS. We have obtained positive results in 7 different readouts in animal models. The characterization of paclitaxel has been described in a +1 manuscript. So the compound has an epigenetic mechanism of action and reduces neuroinflammation and modulates key neuronal plasticity genes. It has a high BBB penetration, and it has been found to be safe and well tolerated in phase 1 and various phase 2 studies. Until now, over 300 subjects have been dosed. As we saw in preclinical models, we found that paclitaxel reduces aggression and agitation in borderline personality disorder, in ADHD, ASD, and AT. Also, it improved the general condition in borderline personality disease patients. These findings were obtained in Reimagine, a phase 2A basket trial in psychiatry. The study was designed as a single-center, single-arm, open-label study. It was an exploratory study which envisages the recruitment of 30 patients. In the end, we recruited 9 borderline personality disease patients, 6 ASD patients, and 8 ADHD patients. So the primary endpoint was safety and tolerability, secondary endpoint reduction of aggression, and other disease-related outcomes. So what we found was that paclitaxel was safe, well tolerated, and it reduced agitation, aggression, and improved the overall status in borderline personality disorder patients. Based on these nice findings, a novel trial was designed, Portico, a phase 2B trial in borderline personality disorder. It is a double-blind, placebo-controlled adaptive design with interim analysis to assess statistical power. We envisage to incorporate around 156 patients. The trial will have 2 primary endpoints: overall clinical BPD improvement and improvement in aggression. CT has been approved in Spain. Identification of sites in other countries is ongoing. The first patient was enrolled in March 21. Similarly, we designed Evolution, a phase 2B trial in schizophrenia. It is a double-blind, placebo-controlled adaptive design with interim analysis to assess statistical power. The primary endpoint of this trial will be efficacy to address schizophrenia negative and cognitive symptoms. LSD1 inhibitors also offer the opportunity to apply the concept of precision medicine to CNS disease. LSD1 is expressed in the CNS and plays a critical role in neurogenesis and the regulation of cortical development. After birth, LSD1 contributes to neuromorphogenesis in the mammalian cortex. LSD1 is the most abundant histone demethylase in the prefrontal cortex, and it localizes in vivo to enhancers and promoters of confirmed CNS disease risk genes. On the other hand, mutations in ZD1A, a protein which exercises kind of the opposite function of LSD1 with respect to the methylation of H3K4. Mutations of SETD1A have been implicated in schizophrenia, developmental disorders like autism spectrum disorder, ASD, and other rare syndromes. Mice with the mutated SETD1A gene recapitulate human schizophrenia symptoms, and Oryzon LSD1 inhibitors reverse these deficiencies. A second example can be found in Kabuki syndrome. Mutations of MLL2 have been implicated in Kabuki syndrome. Mice with the mutated MLL2 gene recapitulate Kabuki patient symptoms like craniofacial abnormalities, growth retardation, and immune dysregulation. Now, LSD1 inhibitors can reverse these deficiencies. Finally, SHANK3 mutations cause ASD, Phelan-McDermid syndrome, and schizophrenia. SHANK3 encodes a protein that is essential for proper functioning of the synapse, the junction between neurons. The majority of people lacking a functional copy of the SHANK3 gene have both autism and severe intellectual disability. Mice with a mutated SHANK3 gene recapitulate the PMS ASD symptoms, and Oryzon LSD1 inhibitors reverse these deficiencies alone or in combination with HDAC2 inhibitors. Thus, LSD1 turns out to be a key target for personalized therapy in CNS disease. Not only can inhibition of LSD1 counteract mutations in genes that are exerting opposing functions, but also LSD1 acts as a downstream molecular hub where a variety of neuronal signaling pathways converge. And acting on LSD1 can compensate distinct independent deficiencies occurring upstream. LSD1 inhibition paves the way for personalized medicine in psychiatry. We have collaborations ongoing with major clinical institutions to inform and prepare clinical studies based on personalized medicine. SETD1A-related schizophrenia is one of the examples. We are collaborating with Columbia University to characterize psychometrically patients carrying mutations in this gene identified in the Amish community. We are also collaborating with Hospital Universitario La Paz in Spain. This institution has characterized over 200 patients carrying mutations in SHANK3, and we will study patients psychometrically as a basis to continue with interventional studies. Finally, and striking an entirely different key, Vafidemstat is also being tested in ESCAPE, a phase 2 trial to prevent ARDS in severe COVID-19 patients. The rationale for ESCAPE was that A, Vafidemstat is safe in the elderly, B, Vafidemstat reduces inflammation and neuroinflammation, and C, LSD1 inhibitors have been reported to reduce the cytokine release syndrome ex vivo in COVID-19 in vitro. A multicenter open-label 2 arms randomized study in combination with best supportive care was designed, which was recruiting 40 plus 20 patients with 6 centers in Madrid and the Barcelona metropolitan areas. The primary endpoint is efficacy of Vafidemstat in preventing ARDS in adult severely ill patients with COVID-19. And the secondary endpoints are reduction of systemic and pulmonary inflammatory biomarkers. Recruitment has finalized and analysis is ongoing. Summarized, at Oryzon, we have a unique dual epigenetic proposition in CNS and oncology. With a personalized medicine approach with multiple shots on goal, we have 2 phase 2 programs. Safety has been proven in over 400 subjects that have been dosed. We are well funded to 2023, and we have different value inflection updates in 21-22. Our programs include oncology with Yadademstat, with the first and best-in-class LSD1 inhibitor in oncology. We have seen over 100 patients dosed. We have robust and durable responses in combination treatment and AML. We have FDA and EMA orphan drug status in AML. We have a SELC trial with ICIN preparation and a dual registration strategy. Then at the CNS level, we have vafidemstat, the first-in-class LSD1 inhibitor in CNS. We have a safe molecule. Over 300 subjects have been treated in several trials. We have seen a reduction of agitation and aggression in psychiatric disorders. We have 2 new phase 2b studies running in 2021 in borderline personality disorder and schizophrenia, and different trials in genetically defined subpopulations are being planned. I would like to thank all the scientists that have collaborated with Oryzon over the years, and I would like to thank volunteers and patients that participated in our clinical trials. I am happy to answer.
So,
if there's any questions for Dr. Tamara, you can use the Q&A tab and the Discuss tab to type it in. I'll give everyone a moment here to think of any questions or comments that you might have, Dr. Tamara.
okay, let me see. Thanks, we can hear you. Well, I think everybody has disconnected, or maybe they're waiting. I just wanted to stress the how different CNS disease. Hello. The treatment of CNS disease is becoming, because over the ages, patients with CNS disease have been submitted to all kind of treatments, from being put on the stake because of their strange behavior, being locked away. Then in more modern times, last century, a lot of people were receiving electroshock therapy and all these treatments, which are basically reflecting that the doctors at each point of time didn't really know what to do with these people. Little by little, we are starting to understand the basis of which can be neurodevelopmental, which can be glitches in the signal transduction pathways, which can be impacts of exposure to certain toxic compounds. We are starting to understand what they are, the effects that they are provoking, and with this understanding, we can start thinking of how to treat patients in a better way. No, we go little by little because knowledge is growing like that. It is not only the work of Oryzon; it's a work of this whole scientific community unraveling these pathways, and this is creating a huge opportunity to start to address these patients in different ways. Not to just sedate them, but to try to kind of balance the unbalanced pathways. And we think that our treatment with our epigenetics compounds is really a good way to go for that.
Absolutely. And you can learn more about their company. We have shared their profile on LinkedIn as well. So I encourage everyone to visit their LinkedIn page. They post continuous updates about their drug development program. And it was a pleasure having you, Dr. Tamara.
Yes. Okay.
With that said, we will be concluding the program. I want to take a moment to thank everyone for joining us today. I'll be doing a quick little raffle. My team is just getting back with more tickets to add. So those that visited the booths today, this is the time. I'll be doing 4 draws, 1 for the Apple Watch winner, 1 for Amazon, another 2 actually for the Uber Eats prizes. So with that said, in my little hat over here, we have The first winner will be for the Apple Watch. So I will select this name over here. James Zhang from GSK, you've won our last Apple Watch 5 series. Come on, email us. We'll ship that over to your address. James Zhang from GSK. The next one that we have will be for a $300 Amazon virtual gift card, which can be redeemed. That prize goes to Paula Puccini, sorry if I mispronounce it, from CHIESI Pharmaceutical. Paula, if you're here, $300 Amazon gift card. Next will be for $200 Amazon gift card. And that will go to Roger Palak of Pfizer. Roger, $200. And the last one for $100 Amazon will go to Mr. Christoph B. Witcher of McComics. Christoph B. Witcher, you are our winner for the $100 Amazon gift card. So for those 4 winners, we'll reach out to you for your prizes. Most importantly, feel free to follow us on LinkedIn, Twitter, and our company page for more updates about our life science events. Again, it was a pleasure having you all join us today. Thank you to all our speakers and attendees. I'll leave this room open if you want to network amongst yourselves, message each other in the lobby, visit the booths, and thank you once again for joining us. Take care. Stay well. Bye.